that wax melts, and the first solution goes, the top solution goes plunking into the bottom solution, and then everything is mixed and ready to go. But here's the key. When it's doing that, it's now over the annealing temperature. It never gets below the annealing temperature. And if it never gets below the annealing temperature until the very end of the cycle, by which time all of your amplification is already done, if it never gets below the annealing temperature, then you're only going to, you don't have to worry about partial matches anymore, because the temperature is high enough to destabilize that hydrogen bonding, so it's not going to happen. But of course, adding that little bit of wax to all 50 or 100 tubes is still kind of a burden. So then, and this is the procedure that we're going to use, a company came up with this idea and said, well, the real problem is the polymerase. If you have the polymerase that cannot add nucleotides until it gets over the annealing temperature, then your problem is solved. You can have everything in the same tube, including the polymerase, and if you have the active site of the polymerase blocked with something, so in this case it's blocked with a little piece of a protein, so if you have your active site blocked with a piece of a protein so that your enzyme does not have activity until after it's over the annealing temperature, then this solves the problem. So we're going to be using polymerase that has a protein blocking the active site, and that protein will not dislodge from the active site until you have heated it to around 95 degrees, which is our denaturation temperature for a couple minutes. And then that piece falls out, and then the polymerase is going to function normally, and then you're going to get that denaturation, annealing, and extension three-step process, and you're going to get a proper addition of the oligonucleotides where they should be binding. So, let's see, that's pretty much the same thing that I was going to tell you. Okay, so now, you're going to then run it on a gel. So after you do the PCR, you're going to be running it on a gel. And there's different kinds of gels. So you might want to take a picture of this. If anyone wants to take a picture of that, this would be a good time. So there's different kinds of gels. There is a polyacrylamide gel, and a polyacrylamide gel will separate things in the range of 10 to 800 nucleotides or base pairs. So things on the smaller side. Things which are in this size range are small RNAs, small DNA restriction fragments, sequencing products. On the other hand, you have agarose gels. And agarose is a somewhat looser matrix compared to the polyacrylamide gel, so it will separate larger fragments, but it works on basically the same principle, is that the DNA or the protein, in the case that you've already done with the earlier in the semester, the protein of the DNA has to get through this matrix solution, whether it's a very thin matrix in the case of polyacrylamide, very small holes, or in the case of agarose, just so many overlapping long strands that it sort of forms a matrix, but it's easier to get through, so it's going to separate larger sized pieces. So agarose gels separate between 300 and 50,000 nucleotides or base pairs. Things which fall in this category are the larger RNAs or the large DNA restriction fragments. And within polyacrylamide and agarose gels, there's two different types of gels. You can have a native gel. A native gel is where you want to keep the DNA or the RNA together. So you'd have double-stranded DNA, or you'd have RNA in its native configuration. And this is done with just a normal salt buffer, like a tris borate with some salt, or a tris acetate buffer, and everything is kept in its native conformation. On the other hand, you can run a denaturing gel. And a denaturing gel is when you want to have single-stranded DNA, or you want to have RNA that's separated on the basis of its size and not its native conformation. And so if you have a polyacrylamide gel and you want to run a denaturing polyacrylamide gel for DNA, you've already experienced denaturing polyacrylamide gels for proteins, but this is for DNA. So if you need a denaturing gel for DNA, you can add 7 to 8 molar urea, which is a chaotropic agent. And this chaotropic agent, the urea, competes for hydrogen bonding with the DNA and the RNA and causes them to become single-stranded and to lose their native conformation. Now, if you're running an agarose gel and you want to do a denaturing agarose gel, if you're doing DNA, you can add sodium hydroxide, basically putting DNA in alkaline conditions. When DNA is in alkaline conditions, it will denature. So you'd run an agarose gel that has a high pH, and your DNA will stay denatured. RNA is a little bit more difficult. RNA, you generally have to use something like methylmercury, which is extremely toxic and deadly even if you're not wearing the right type of gloves, or glyoxal, which can cause severe allergic reactions in people who are allergic to it, or formaldehyde, which is a potential carcinogen. So none of these choices are good. So if you have to run a denaturing RNA gel, you're probably using formaldehyde as the least bad choice there. But even that comes with lots of environmental health and safety regulations for working with that stuff. So you're running a gel. Get my next slide here. What is it? Again, you have a picture of this in your lab manual. On page, I don't know, page 94, page 94, 93, 94 of your lab manual. Okay. It's hard to sit and listen to somebody for a couple hours, isn't it?
No.
My butt gets sore too, in case you're wondering. Okay, so let's say you have some long pieces of DNA, 45,000 base pairs, 28,000 base pairs. You cut them with a restriction enzyme, so you have a bunch of different smaller sized fragments, and you're going to run them on an agarose gel. This is just a for example. So you run them on the agarose gel, and remember you had two different sizes making a bunch of different sized pieces. And unlike what you did in the aldolase experiment when you ran the SDS gel, in that one you calculated the RF because nothing ran before the dye front. When you are dealing with DNA gels, polyacrylamide or agarose gels with DNA, you cannot use an RF because it is possible for small fragments of DNA to run before the dye, and it's not separating on the same basis anyway. That it doesn't have glycine in there, so that's not happening at all in the same way. It's not an SDS denaturing gel at all. So please do not confuse the two. So let's say you added fragments from DNA piece A that was cut up, and fragments from DNA piece B, and you ran them on the gel. And then you measure, and you can say, okay, well, B started out here, and I can measure how far did this piece run, how far did this piece run, how far did that piece run, and I know what size they are. And you can make a graph out of that. Kind of looks like this. And again, this is in your lab manual. So there'll be a part where it's fairly linear if you're using sort of a medium percentage gel. And remember, one of the things you learned in your aldolase experiment, you can choose all different percentages of gels. And you choose different percentages of gels based on what size protein, in that case, you were needing to separate. And the same thing holds true for DNA. that holds true for protein. If you needed to separate out, and you can see it's a little bit more difficult to separate out, say 220 from 350. They're at the smaller end of what can be done in an agarose gel. 350, 600, you're really not getting any linearity in here at all. If you needed to separate out efficiently a 350 from a 500, from a 600, etc., you might want to make sure it's more linear in this region. So you would choose a higher percentage gel, because a higher percentage gel is going to separate out the lower molecular weight pieces better. And it does that. A higher percentage gel, everything has a harder time getting through the gel, but the smaller the pieces are, the easier time they're going to have getting through the gel. So a higher percentage is going to allow you to separate out all those small fragments, where all the large fragments, 3,000 and up, they're all going to be just basically in a big lump at the top and not separated out well at all. On the other hand, say you needed to separate out efficiently 6,000 from 12,000 from 19,000. Well, in that case, you would use a lower percentage gel, because a lower percentage gel is going to allow you to separate out the higher molecular weight fragments better. Because when there's larger holes, everything goes through faster, and the smaller pieces are all going to go through, boof, all at one time, so they're not going to separate out very well, but you're going to get some separation in the larger size fragments. So you can choose your percentage of gel based on what you need to be separating out, and it's very flexible. And this holds true for agarose, holds true for a basic acrylamide, holds true for STS page gel. It's the same rule for all of them. It operates exactly the same. Okay, so it's back neater. So in this experiment, you're going to be working with biological material, and I actually developed this is the experiment that I developed for the class, because I was very interested in forensic science. I don't know whether you guys know, but I teach forensic science here. And when I was getting into forensic science, and I'd taken a bunch of classes and stuff, I decided I needed more practical experience. And so to get practical experience, I took one summer off and I worked at a paternity testing lab. And I took another summer off and I did an internship with the San Diego Police Department, learning forensic biology from them. And so I put together the different techniques that they were using in the labs to make this experiment. So the paternity testing lab was doing the DNA IQ system that we're going to use with the magnetic bead technology, and the police department will PCR up short tandem repeats. So this all comes from biological evidence. And biological evidence can be blood that's left behind in a crime scene. It can be semen that's in a rape case. It can be saliva. In a lot of rape cases, the victim might have their breast licked or something like that. It can be urine. There was one case where this burglar, and you're going to break into somebody's house, you might be a little nervous. Well, this burglar was a nervous peer, and so he had a habit of peeing against the side of the house that he was going to break into. So eventually the police figured this out, and they were able to get his epithelial cells, because you shed epithelial cells everywhere. You shed them when you urinate, you shed them when you poop, you shed them in your saliva. So you are discarding, you shed them when you touch things. So they were able to get his epithelial cells and get a genotype basically for him. So there was another burglar who, as his calling card, left poop in somebody's living room. So it's kind of like a power play. So he'd be like, I was here, and he would take a dump in the middle of somebody's living room. But what he didn't realize is the police can carefully get DNA from poop. Now, keep in mind that your poop is what you've eaten. So you have DNA from your food in your poop. You have DNA from bacteria in your intestines, in your poop. But on the outside of your poop, as it slides along your colon, you're picking up a lot of epithelial cells from your colon. So a very careful extraction of just the outside of the poop gives you an enriched layer of epithelial cells, and then they can get, a human profile. And you're amplifying bacteria and food, but remember, they're designing this PCR oligonucleotide so they're only going to bind to human DNA. So it doesn't matter if you get other kinds of DNA in there, you're amplifying only the human DNA. So you're going to have a source of biological evidence. In your case, you're going to be taking epithelial cells from your cheek to be able to amplify those. So initially, there were 13 locations on the DNA that the police would amplify. And these locations on the DNA had quite a bit of research that went into them. You have in your DNA coding regions that code for a gene, and you have non-coding regions. And the non-coding regions of your DNA are free to mutate without killing people. And so over many years, your non-coding regions of your DNA has mutated, and repeating segments of DNA have come to be through, it's an enzymatic process called stutter, and I'm not gonna go into the whys and wherefores because this is not a forensic science class, but you will get repeating sections of the DNA. So one of the handouts I gave you shows a little sequence of DNA, and it shows you AATG, 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 AATG repeated a number of times. So that is a short tandem repeat. Short because they're usually 4 to 5 bases long. Tandem because they're right next to each other. Repeat it over and over and over again. So what they have done, scientists since the 1980s, scientists have been developing this methodology since the 1980s. And what they've done is they've found places on the DNA which have a great deal of genetic variation from person to person. So, for example, if you are looking at blood types, the vast majority of people are type O positive. And a heck of a lot of people are type A positive. So if you find very few people are type B or type AB. So if you find type O positive blood at a crime scene, that's not very discriminatory in terms of being able to rule out who committed the crime. So there was a need in police work to be able to get away from just simply typing the DNA, typing the blood, to how can we use people's DNA to get a genetic profile. So the research that went into this found places in the DNA that had variation among people, because just like blood types, if almost everybody is type O or type A, and you find type O, it's pretty useless. But if you are looking at a place in the DNA, and you can say, okay, well, a lot of people have 8 repeats, but some people have 7, some people have 6, some people have 9, some people have 10. And maybe 10% of the people have 6, and 10%, and 20% have 7, and 30% have 8 repeats, one after the other. But then, some people have, percentage have 9, and percentage have 10, etc. So now you're saying, okay, if we find 6 repeats at this particular location on the DNA, we can use this to rule out anybody that has 7 or 8 or 9, etc. So you can use statistics to do this, to say how likely is it that you would have 6 repeats on this piece of DNA that we are looking at. So initially, the police identified 13 different locations on the DNA, and they were able to get, using statistics, they were able to get and say, well, the chances of having say, TPOX, TPOX, or the D5S518, there's maybe only a 10% chance that anybody would match this pattern. Or at D5, maybe there's a 10% chance that anybody would match that pattern. And so when you start applying statistics, if you've checked 13 different locations on the DNA, it's the probability of this times the probability of this times the probability of that, and that gives you a much lower probability. So when they started looking at these original 13 locations, they were able to fairly easily get the probability of anybody having the same pattern of DNA is 1 in 10 billion, which was great initially. But now, as the population of the Earth is over 7 billion and climbing, eventually you're going to have some defense attorney that tells the jury the population is, the probability of someone else having the same exact pattern is 1 in 10 billion. And the population of the Earth is almost 1 in 10 billion. Therefore, there is another person on Earth that has the exact same genetic pattern as my client, and that, ladies and gentlemen of the jury, is the person that committed this crime. And for the jury, it doesn't matter how likely it is that person happens to be in San Diego at this exact time, because juries don't understand science. And if you have a good lawyer who can twist the science a little bit, you can get juries to believe a lot of things. So the police department eventually said, crap, 13 is not enough. We're going to go to 24 locations on the DNA. So now they have 24 different locations that they test on the DNA, all of them doing PCR. So they are PCRing up every single one of these locations, and all in the same time, and all in a single tube. So they have advanced the process so that they can do PCR on 24 different in the crime lab. They will isolate a sample of DNA from a crime scene. Doesn't require that many cells. You leave enough cells behind when you touch something with your finger to give a genetic profile now. They need about 10 cells. And so it's very possible that you touch something and you've left 10 cells behind at a crime scene. So they will find something that someone has potentially touched. They will do a genomic DNA isolation. They will do PCR on the 24 different locations, and then they will start doing statistics. And we're going to learn how to do statistics on Monday, so I'm not going to get into it here. Other than to note, they've now got locations on every single one of the 22 chromosomes to avoid trying to get the maximum flexibility. And how they're able to tell whether it's male or female DNA that's been left behind at a crime scene is from the amylogenin gene. The amylogenin gene on the X chromosome is one particular size, and on the Y chromosome it's a different size. Now for everybody who has an X chromosome, it's all the same size. And for everybody who has a Y chromosome, it's all the same size. But they're not needing to discriminate between what the size is. They're just saying, do we have two different sizes when we PCR up the amylogenin gene? And if we have two different sizes, one comes from the X and one comes from the Y, and it's a male. And if you only have a single size of the amylogenin gene, it's because there are two X chromosomes, and so they both are going to give the exact same size. So if you only have one band in the amylogenin gene, it's going to be a female because it's XX. There, of course, are XXY cases and weird things like that, but for the most part, we're just going to confine ourselves to XX and XY because that's a whole bunch of different chapters in a book, going beyond that. Okay, so they have weird names to them. They have weird names like TPOX and CSF1PO. So initially, when people were developing all these short tandem repeats, everyone was naming them according to their own little nomenclature. And finally, they got together and said, come on, guys, we need a consistent naming system among all of these. And so that's when they came up with things like D5S, 818 or whatever. So they said D for DNA, 5, it's on the 5th chromosome, S, 18, S518 or whatever, that's the particular location on the chromosome where this particular short tandem repeat is found. So now I want to talk about this slide. Okay, so we've talked about PCR. And so let's say someone's looking at chromosome number 5. And they have on chromosome number 5, just outside the potential different number of repeats that everyone has. Because remember, there are tens of thousands of potential places that have repeats. So these were specifically picked for having wide genetic variability in how many people had each of these number of repeats, and for having very consistent locations where the primers can bind. So when you are binding primers outside of these repeats, the repeat region is variable between the samples. So if we're looking at chromosome number 5, you might have 6 repeats, 7 repeats, 8 repeats, 9 repeats, and they're going to be one after the other, but 8 repeats is bigger than 7 repeats. It's going to be a little larger. But the important thing is the place where the primers are binding has to be constant. So for each individual location on the DNA, chromosome number 5, they had to test to make sure that everybody has the exact same sequence on chromosome number 5, so they can make oligonucleotides, which are going to bind to every single person's DNA and amplify in between those primers. Now, you're not amplifying large pieces of the DNA. You're amplifying usually between 300 to 600 bases. So all of these fragments are very small. They're not sequencing the DNA at all. Sequencing is done by companies like 23andMe and Ancestry, where they're looking at single nucleotide polymorphisms in the DNA, but they're not actually, the police department does not sequence the DNA. They're only interested in how long it is. So you might have inherited a 7 from your mother and an 8 from your father, or you might have inherited an 8 from your mother and a 7 from your father. So there's 2 different ways that you can be a 7 and an 8. And that's homozygous. And the 2 different ways is going to play in when we talk about statistics on the Monday after spring break. But basically, you're at each location that they're PCRing, you're either going to be homozygous or heterozygous. If you inherited 2 different numbers of repeats from your parents, they're going to be different sizes, and they can be resolved on a gel because they're going to be slightly different sizes. Something with 8 repeats is going to be a little larger than something with 7 repeats. And if it's homozygous, it meant maybe you got an 8 from your mother and an 8 from your father. So you got the same number of repeats from both parents. So when you are doing this, that's one location on the DNA. But I've said they're doing 24 different PCR reactions all at the same time. So how are they able to do 24 different PCR reactions all in one tube and still be able to separate all of those out? So one thing they do is they change the distance where the primers are binding. You can have the primers binding very close to where your repeats are, and if the primers are binding very close to where your repeats are, you're going to get smaller size ranges of fragments. And if your primers are binding farther away from where your repeats are, you're going to get larger sized fragments. And they can be resolved on a gel because they're going to be different size ranges. So now, I want you to look at this picture that I gave everybody. We're going to talk about that. So you're looking at this picture right here. Okay, so look at panel C that's on the right-hand side. And panel C has a genetic profile for 6 people. So see the numbers 1, 2, 3, 4, 5, and 6. Those are 6 different people that they have done PCR on their DNA. Any lane which has an L has a bunch of size markers. And those size markers are specifically designed to tell them and say, okay, if you have, let's say TPOX. If you have TPOX, if you have 6 repeats, it's going to be this size, the same size as that lowest band in the L on TPOX. For all the L's. All the L's are exactly the same. So the L's to the left, the L's to the right, the L's in the middle, they're exactly the same set of markers. You have markers, 2 people DNA, markers, 2 people DNA, markers, 2 people DNA, so that you always have someone to exactly compare it to. So let's say person number 6 on TPOX, and you wanted to say, okay, how many repeats, if we are amplifying the TPOX location in their genomic DNA, how many repeats does that person have? So what does it line up with? So you can see, 6, 7, 8, 9, 10, 11, 12, 13. So what does person number 6 in TPOX line up with? 8. So they would have, is that homozygous or heterozygous? How many bands do you see? If it's 1, is it homozygous or heterozygous? It's homozygous. So this means they inherited an 8 from their mother and an 8 repeat from their father. Okay? So both parents contributed the exact same number of repeats. And you can, as you look across person number 1 and 2 and 3 and 4 and 5 and 6, you can see 8's pretty common. A lot of people have 8 repeats of TPOX. I have 8 repeats of TPOX. Now let's look at TH01. Let's look at person number 6 in TH01. So same person number 6 on the right-hand side, just down 1 to TH01. How many repeats does person number 6 have at TH01? 6 and 8. So does anybody not understand, this is important, does anybody not understand why it's a 6 and an 8? Don't be afraid to holler. Okay, so that's good. You got to be brave. Okay, so look carefully at person number 6 at TH01, and you can see that size marker. And if you see that size marker, it goes from 5 to 11. And so you have 5, 6, 7, 8, 9, 10, 11. And so you can see person number 6 has little bands next to the 6 and the 8 of the size markers. Do you see that? Okay, so this tells you they're a 6 and an 8. Does that work out for you too? Okay, so then you can say person number 6 is a 6, 8 at this location. And then you can go on and you can do CSF1PO for person number 6, and you can do VWA for person number 6, and you can do, because you are changing the distance that the primers are from the repeating unit of DNA, you're going to get a different size range on the gel. So you can separate out on this gel, you can separate out 4 different size ranges. But wait, she said that it was possible to do 24 different ones all in the same tube and all in the same gel. How is this possible? Well, now look over to the left, to that little black figure. What color are the CSF1PO, the TPOX, the TH01, and the VWA in? Okay, so they're all in red. So that's because they all have a red fluorescent marker on the ends, added to the ends of their oligonucleotide. So you can tell the computer, show me only the red, or show me only the green. So you can be running all of these bands at the exact same time. You can be running 24 different reactions, but if these 24 different reactions have a bunch of different fluorescent markers attached to them, then you can you can just see a couple at a time. So you can have the red where you can see the panel on the right. You can have the green where you can see the center panel. And there's blue, and there's yellow. There's a bunch of different kinds of fluorophores that you can add to your oligonucleotides, and then you can amplify all 24 at the same time and just see a couple of them one at a time to be able to look at them along with the appropriate size markers. Because if you look, the size markers come in red and green also. So the size markers, for VWA will show up if you say, show me red only. And you'll get a completely different type of size marker if you're looking at the greens, which goes for the D6, which is in that exact same size range, but now you're telling the computer, show me only the green. So this idea is called multiplexing, of doing multiple PCR reactions, far more than one PCR reaction at one time. And so when we multiplex, we're actually going to be using CSF1PO, TPOX, and THO1. We're going to be amplifying all three at the exact same time on your genomic DNA that you will have isolated from your genetic sample. So I think, yes?
Can you explain how we know if it's homozygous or heterozygous?
So, okay, excellent. So if you look at TPOX and look at person number 6 in TPOX, how many bands do you see for person number 6 in TPOX? One. So by definition, it's homozygous, because if you have inherited 8 repeats from your mother and 8 repeats from your father, they're going to be exactly the same size. And so we call that homozygous. They're the same size and they cannot be resolved. You don't know. You're just doing the size. You don't know, which came from your mother, which came from your father. It doesn't matter. You're homozygous. But versus THO1, you see 2 different bands there. By definition, you're heterozygous. Okay, I think I will follow all this up on the Monday after spring break.